Imagine you are the secretary of the BKS and you have to make 3 particular students sign a form. How would you contact them? Naturally, no one would send the form in question to every student of this school and say they have to complete it in order to make sure that the 3 concerned people will do so, because this would cause a lot of commotion and had significant side effects. First, the secretariat was drowned in useless paper. Second, all the other students were quite annoyed about having to complete a form on something that doesn't even concern them. And third, a lot of paper was wasted. A much more efficient approach would be to find out in which classes the 3 concerned students are and just send the form to them, or, of course, to contact only the people in question. Everything else appears rather silly. But until now, drug-based treatment of central nervous system diseases has looked more or less like sending a form to 1,000 people to reach 3. These examples might seem incomparable, but they have some things in common. The 3 target students are the equivalent of the brain area where a disease occurs and needs treatment. The other 997 students compare to the rest of the body. The form letters correspond to the medication needed and the secretary to the doctor. Now, why did physicians choose such an inefficient approach to treat brain diseases? Because they had no other choice. Until now, the only possibility to deliver a drug exactly to its target has been via surgery. Quite a riskful thing when such an easily damaged and essential organ like the brain is involved. The only remaining option is to inject a medicine systemically, which means that it circulates in the blood vessels in the entire body and eventually reaches its target. But the majority of the injected substance goes astray. There occurs another problem. Almost every receptor is used for several functions. So maybe we have one molecule that can bind to its corresponding receptor in an arterial cell and cause an increase of blood pressure. But if this same molecule reaches the same receptor, but this time on a gland cell, it may cause the production of another hormone with a variety of side effects. So, if scientists find out which molecule is responsible for a certain job and even find a way to synthesize it and develop a medication, it might fulfill its task, but also many others. This means, by sending the form to every student, the concerned ones will surely obtain it, but also a whole lot of other people who don't need it. But if it is the only way to treat a disease, the side effects are the lesser evil. Another obstacle we have to deal with in the brain specifically is the blood-brain barrier. This is a semi-permeable membrane that surrounds every blood vessel in the brain. Its job is to keep toxins and everything harmful away from the sensitive neurons, but as it works well, it also keeps drugs out. Some molecules pass it, mainly small and electrically uncharged ones, so medications need to fit the scheme. It can be forced to open, for example with ultrasound, which then allows also other molecules to enter neuronal tissue. But this might cause serious problems because it allows many substances to interfere with neurons. To avoid complications and make medical treatment easier, scientists have been investigating the field of medicament delivery for quite a while, and recently a team from the ETH Zurich has found a promising approach. They put a model drug into fat bubbles, so-called liposomes. Then they tied the liposomes to bigger liposomes filled with perfluorobutane, a liquid which is especially sensitive for ultrasound. This was then injected systemically into anesthetized rats. With a novel focal ultrasound sequence, the investigation team managed to concentrate the liposomes in a specific brain region they wanted to modulate. They also found a different ultrasound sequence which destroys the smaller liposomes and causes them to release the drug at exactly the spot wanted. In our example, this corresponds to choosing only the concerned people to send the form to. The idea has existed before, but the team of the ETH has improved it. Their method required ultrasound pressures many times lower than in former experiments, which minimizes off-target effects. With their ultrasound sequence, the blood-brain barrier had also stayed intact. As the whole method involving ultrasound and liposomes delivers a drug specifically to its target, there are only little side effects, because it won't reach anything else. They are even less since the amount of drug needed is a lot smaller. These two effects also enable the use of substances which have not been used as medicaments because they had a toxic effect in larger doses, but if they are used frugally, they can have therapeutic effect but no other. But there is still a long way until this method can commonly be used in treatment of neurological diseases, as it first has to pass various safety checks.